The rest of this unit, Chapter 12 and Chapter 13, we're going to be looking at host defenses. This is one of my favorite sections of microbiology, other than learning about the critters themselves, because we're learning about our immune system, all right? So host defenses are a multi-level network of what we call innate. Innate means you are born with it. Non-specific protections, okay? So innate, kind of broad spectrum protection. This is not what we think of as immunity, okay? Then we have our adaptive specific protections, more commonly referred to as the sorry, I'm getting ahead of myself. The adaptive specific protections we're going to have, and this is where our what we think of as our immune system is going to come into play here, okay? So we're going to break this down into 3 lines or levels of defense or protection against infection, our first, second, and third line of defense, okay? So we categorize them primarily, and I've already said this, but the host defenses as either innate, which is non-specific, born with it, or acquired specific defenses that you kind of have to pick up along the way. And so we're going to look, there's going to be a chart, and I'm going to bring this chart up over and over again. I would definitely learn this particular flow chart and commit it to memory, because that kind of gives you the overall broad view of this particular portion of this unit. All right, so here's that flow chart I was telling you about. I'm going to give you a lot of information here, and then we're going to come back to this, okay? So, like I said, our defenses are classified into one of 2 general categories. We have our innate abilities. You know, you're born with something, it's innate. These are our non-specific, so they're kind of broad spectrum, meaning they fight many different types of pathogens, not something in particular, okay? So these you're born with, all right? Not what we normally think of, especially the first line of defense, not what we normally think of as our immune system, okay? And then we are going to have our acquired or specific. This is where our things that we think of as our immune system lie, okay? And then we have some that lie in between the second and third line of defense there. But and so these are going to be things that we acquire that we pick up along the way. All right. And so then these can be further divided down into our first line of defense, our second line of defense, and our third line of defense. Okay. And so this is going to be the most complex and responsible for very specific immunity against specific pathogens. Okay. So the first line of defense is where we're going to start over here. This is going to be a unique type of surface protection composed of anatomical and physiological barriers. And you've already heard me talking about this, okay? These keep microbes from getting through to sterile body compartments. We're going to have physical barriers, chemical barriers, and even some genetic components. Physical barriers are going to be things like unbroken skin, mucous membrane, hairs, tears, flow of fluids is what I like to call those. The second line of defense is cellular and chemical, and that's going to come immediately into play once the infectious agent gets past the surface or past that first line of defense. You can think of the first line of defense kind of like a big gate with razor wire all around, trying to keep you out, okay? That second line of defenses, we're going to have those things like phagocytosis. We're going to have inflammation and fever, antimicrobial proteins, other ways that our body tries to fight off microbes in a general, not necessarily specific manner. Then our third line of defense is going to include very specific host defenses that must be developed uniquely for each particular microbe through the action of our white blood cells, particularly our B cells and our T cells, our T lymphocytes, which develop actively through an infection or passively through things like maternal antibodies. And this is where we get our memory responses and where we can actually have immunity to certain things. Like we get the chicken pox, we develop antibodies to the chicken pox, and so we are no longer able to get it, is the idea there. All right, so the first line of defense, what part of immunity is that? Innate or acquired? That's innate, okay? Non-specific. So this is any barrier that blocks invasion at the portal of entry. It limits access to the internal tissues of the body. Okay, this is that wall keeping things out. It's not considered an immune response. It doesn't involve recognition of foreign substances, very general, and it's just kind of designed to keep everyone out. So the second line of defense is going to be more cellular. It's part of the innate defenses, still very non-specific, think kind of broad spectrum. So this is an internalized system of protective cells and fluids, includes inflammation and phagocytosis. It acts very quickly on a very systemic overall organismal level. And this again is activated once the first line of defense is overcome. And we're going to go into much more detail on all these things, okay? The third line of defense is acquired on an individual basis. This is what you're going to pick up as you go, as each particular substance is encountered by a lymphocyte, okay? So the reaction with each different microbe produces a unique protective substance and gives us that long-term immunity. And our body is really very efficient at having, and actually we already have, a particular match for anything we can come into contact with, a particular lymphocyte, I should say, that can match any particular pattern on an antigen, which we're going to get into, that we can come in contact with, which is a huge number of lymphocytes or white blood cells, okay? The issue is there's usually only one of those, and we have to go through this process of copying them when we actually encounter something for the first time. Most of our defenses overlap, okay, and they're redundant or kind of repeated in some of their effects, all right? The assault force makes the survival of invading microbes unlikely, okay? But of course, they're crafty little critters and they get through there. And so we have immunology, which is the study of all features of the body's second and third lines of defense, okay? The study of the body's response to particular infectious agents, allergies, study of allergies and cancer. And so there's many different functions here or mandates of the immune system. It is responsible for surveillance, search, recognize, and destroys. These are the three functions, okay? White blood cells are very sensitive, basically of touch, and they actually feel for these surface markers as they travel through tissues. The surface markers on other molecules is what I'm saying, or other microbes, so to speak, other tissues and cells. They help determine what we call self and what we call non-self. So if something is labeled or tagged, I should say, with these surface markers as self, the body says, you belong here. You are part of me, your family, you stay all you want, okay? If it is labeled as non-self, then your body is going to attack, all right? The attack force is going to be sent in, all right? That's going to be non-self. So when self is recognized, there is no immune response that occurs. When non-self is detected, a reaction to destroy is mounted, okay? The most common method of destruction is going to be phagocytosis. This is also where autoimmune diseases come into play. Your cells sometimes get tagged as non-self due to association with a virus or some other things, and so your body no longer recognizes its little name tag as self, and it thinks it's non-self, and so it attacks cells that are actually supposed to be there. The immune system doesn't exist as a standalone organ. It's a large, complex network of fluid cells and other aspects that do work together to screen the body for harmful substances. These are what we call antigens. These are the markers, okay? An antigen is any foreign substance that stimulates a specific immune response. This is often a microbe, but it doesn't have to be. It can be something like pollen, all right? It's going to have proteins and a few sugars, allow cells of the immune system to identify if it has been newly discovered. And if it's a cell that's going to pose a threat, then it will be marked for destruction. So all of the cells that we encounter, they have that unique form of ID tag. It's a mix of those macromolecules on the surface that the immune system uses to determine if they are foreign or not, self or non-self, okay? So white blood cells constantly move throughout the body looking for potential pathogens. They're scouting, okay? And so they want to recognize self cells. They can differentiate them from in a foreign material, which is non-self, and one that belongs there. The ability to evaluate these molecules as self or non-self is central and vital to functioning of the immune system. As I said earlier, many of the autoimmune disorders are a result of the immune system mistakenly attacking your body's own organisms or joints, that type of thing, because they have somehow gotten associated with non-self and mislabeled. So host cells, and the host, of course, is the one being affected, we have these pathogen-associated molecular patterns, PAMPs, or pattern recognition receptors, PRRs. And so the host cells use PRRs to recognize the PAMPs. Okay, so PRR is the host cell, PAMP is the microbe. It has an M, so that might help you remember that one is microbe. So these non-self proteins that are not harmful are going to be recognized as such, an immune system is going to not react. Okay, but these markers are what we're talking about. So the body is partitioned or divided off into several fluid-filled spaces, okay? We have the intracellular environment versus the extracellular environment, okay? And so intracellular inside cells, extracellular outside cells, and you can kind of see here that this is complex. We've got blood vessels. You're going to have inside the blood vessel, outside the blood vessel, inside the white blood cell, outside the white blood cell, okay? Intra means inside, extra means outside, all right? And so we have intracellular, extracellular, lymphatic, circulatory, and cerebrospinal. All right. All of these are physically separated, but they do have connections. Okay. So for immunity to work, the activities in one compartment must communicate with the others. For effective responses, the activities have to be communicating. Okay. At the microscopic level, we have clusters of tissues that are in direct contact with what we call the mononuclear phagocyte system and the extracellular fluid. This association, or just close, being physically close, allow the cells here to move in and into the blood and the lymphatic vessels here. All right. So it's important to understand the role of the circulatory and especially the lymphatic system. Okay, so here on the left you can see the circulatory system, and then on the right you can see the lymphatic system, and so they run very parallel. So this is a complex network of vessels, cells, and specialized accessory organs. All right. The function of the lymphatic system is to transport lymph through an increasingly larger tributary system of vessels and lymph nodes, which are going to lead to major vessels that will drain back to the circulatory system. In the circulatory system, the body compartments are going to be screened by circulating white blood cells, okay? The lymphatic system is going to be that branching network of vessels that extend into most body areas. You can see all the way down to the tips of your fingers and toes. You can see there is a higher density of lymphatic vessels in the dead end area, the hands and feet, breasts. These are frequent contact points for infections. Other lymphatic organs in the lymph nodes, spleen, or what we call gut-associated tissues, thymus gland, and the tonsils. On the far right is kind of a comparison of the generalized circulation of the two. Although lymphatic vessels are parallel to regular circulation, it's important to note that they only travel one direction. Okay, they don't have that cyclic nature of the blood. There is a direct connection between the two. Circulations occur at points near the heart, where we have large lymphatic ducts that are going to empty their fluid into veins. All right, so the lymphatic system gives that route of return for that extracellular fluid to go back to the circulatory system. It can be a drain off system for the inflammatory response. And then, of course, it is going to act with or as a vital part sorry, of our immune system to render surveillance, recognize and protect against foreign invaders using lymphocytes, phagocytes, and antibodies. It's going to begin as tiny capillaries that transport lymph fluid through the system of vessels and filters, the lymph nodes, and lead to major vessels that drain back into the regular circulatory system. Lymphatic fluid, or lymph, is formed when certain blood components move out of blood vessels into the extracellular spaces and diffuse or migrate into lymphatic capillaries. It's going to transport many white blood cells, fats, cell debris, and infectious agents that have gained access to lymphatic tissue spaces. All right, so lymphatic vessels, these guys don't have high pressure, so they're similar to thin wall veins. They accompany blood capillaries, you can see here very closely, to facilitate ease of exchange. All right. There are two important differences to note between the lymphatic system and the bloodstream. The first is the one I've already stated. The flow of lymph is one direction. Lymph moves from the extremities towards the heart, returns to the bloodstream through the thoracic duct and the subclavian vein. Second is how it flows, okay? So blood uses the heart as a pump, so it pumps it through. It does use some skeletal muscle contraction, but the primary source of movement is the heart. Lymph is moved only using skeletal muscle contraction. So we have some different lymphatic organs. We have the primary organs and the secondary. Where immune cells are birthed and where they mature are the primary lymphatic organs. Where they are activated and function are the secondary lymphatic organs. All blood cells are going to originate in the bone marrow, even the B and T lymphocyte as precursor cells. They're going to move then to the thymus and spleen once they express markers that identify them as B or T cells. So lymph nodes are small bean-shaped organs. They have a capsule around them. They are stationed in clusters along lymphatic channels and large blood vessels of the thoracic and abdominal cavities. You'll find major clusters or aggregations of nodes in loose connective tissue of these particular areas that you're probably familiar with. The axillary nodes, axilla or armpit, the groin in the inguinal area, so those are the inguinal nodes, and then the neck and cervical nodes here. Okay, so their function is filtration, and of course providing immune cells. Enlargement of lymph nodes can be very helpful and provide physicians with important clues to a patient's condition, as it can indicate a local or a systemic illness. So here we have the spleen located at the end of the pancreas. Here, this serves as a filter for blood. It removes worn out red blood cells, but it also filters pathogens from the blood for phagocytosis and macrophages. Adults that do not have a spleen, that's what asplenic means, A means without, can live pretty normally, but children that have their spleen removed are severely immunocompromised, okay? And so that's important. So we have some lymph tissues, some other lymphoid tissues here, the tonsils. This is an active source of lymphocytes in the pharynx. You know, people tend to think that the tonsils have no function, or we were kind of taught that at one point, but they actually do. Pregnant women, if they are lactating or producing milk, their breast also become a temporary site of antibody-producing lymphoid tissue. The appendix, basically the same as the tonsils, except it doesn't feed the pharynx there. It's going to, though, be an active source of lymphocyte. We have lacteals, which are ducts, and then we have what we call Peyer's patches. These are kind of interesting. These are little clusters of lymphocytes that are found in the ileum of the small intestines. Right, so whole blood is going to be composed of blood cells, which is the solid elements, suspended in the plasma, which is a clear yellowish fluid. Plasma is the liquid portion which suspends the blood cells. It is mostly water. It contains proteins like albumin. It contains globulins, fibrinogen. Um, Serum is basically the same as plasma, except it is the clear fluid that you can get with out blood clotting factors. Okay, this is used in immune testing and therapy. Hematopoiesis is going to be the production of blood cells, white blood cells, red blood cells, and platelets. Blood also contains stem cells. Well, stem cells are actually precursor cells. This is the precursor for new blood cells. These are categorized based on how they differentiate, who they grow up to be. We have what we call pluripotent cells that can become any type of cell. So we have pluripotent blood cells. So we have this precursor cell that is pluripotent that can become any type of blood cell, a red blood cell, a white blood cell, a platelet, okay? We also have totipotent stem cells, and this is like an embryonic stem cell that can actually differentiate into any type of cell, okay? So just kind of important to help understand the difference there. Differentiation is the process where which they change into different types, okay? We have white blood cells, which are leukocytes, leuko meaning white, cyte meaning cell. We have granulocytes and agranulocytes, depending on whether or not we can see granules when we stain them. Okay, these cells are vitally important to the nonspecific and specific immunity. We will work on identifying them. The granulocytes are going to be neutrophils, eosinophils, and basophils. Okay, so NEB, NEB. Agranulocytes are lymphocytes and monocytes. There's a little acronym to remember the 5 types of white blood cells. Never let monkeys eat bananas. So neutrophil, lymphocyte, monocyte, eosinophil, basophil. Okay. So knowing the types of blood cells, being able to identify them, which you will do in the white blood cell differentiation lab, and knowing when they will be elevated is really important. So for instance, if you do a CBC and you find that eosinophils are elevated, that can mean that you have a parasite infection, because eosinophils will be elevated if you have a parasite. Okay, so this information is really, really relevant and important. All cells in the blood do originate from that hematopoietic stem cell in the bone marrow. Leukocytes, we have many different types. And then, of course, we have our red blood cells and our platelets, which are shown down here. Okay. Undifferentiated stem cells differentiate or change. They grow up, give rise to several cell lines that become specialized until mature cells are then released into circulation. All right, so mature white blood cells are going to be our leukocytes, and here they are divided into our agranulocytes and our granulocytes. All right, monocytes here, when they leave circulation, all right, when they leave the bloodstream, they will differentiate or change or mature into a macrophage or a dendritic cell. It's important to understand that. This is the classic appearance of a monocyte, okay? Then here's our lymphocytes. We have several different types here. We have B cells, we have T cells, we have natural killer cells, and then we have our granulocytes over here. Okay, so remember, we have monocyte and lymphocyte as our basic classification here of agranulocytes. Over here, we have the granulocytes. We have our neutrophils. These guys live a short period of time. These are important to do phagocytosis, okay? Then we have our basophils. These are really important for inflammation. Eosinophils, as I already said, are active with protozoa helmets. Also allergic reactions, so inflammatory. And mast cells here are this kind of extra. These are similar to basophils. These guys trigger local inflammatory reactions like allergic reactions here, okay, from allergics. symptoms from allergic reactions. All right. Trying to get this off. The other one that can do phagocytosis is the macrophage, okay? So it's really important you know that. You can have a macrophage and a neutrophil are the two that are really productive in phagocytosis. All right, good little review question here. Which of the following is a filter for worn out red blood cells? All right, A, thymus, B, liver, C, spleen, D, lymph nodes, or E, kidney? Pause. Get your answer. It is the spleen, C. All right, so I kind of already mentioned cytokines. Cytokines are basically chemical messengers. They're also sometimes proteins that help to influence our defense response and allow immune cells to communicate. We can have that cytokine storm that I mentioned as seen in the flu or our COVID-19 as a result of a microbe causing an overreaction of the host immune system. Hundreds of small active molecules are secreted. They regulate, stimulate, suppress, and otherwise control many aspects of cell development, inflammation, and immunity. We have many different types of cytokines, okay? These guys are going to be produced by monocytes, lymphocytes, macrophages, fibroblasts, platelets, other things, okay? The effects can be localized, so a small area, or overall systemic, and that's where it can become dangerous. They can last for a very short time or be long-term, specific or non-specific, and their function is to be protective, but if they get out of bounds and overreactive, then it can be pathologic. Okay, so here's just a good little chart here of some different types of cytokines. Their examples like interleukin or tumor necrosis factor, serotonin, histamine, and you'll hear me talk about erythropoietin. We'll talk about all of these, okay? Where they are released from and where the target cell is. Some of these are particularly important. Well, let's just kind of go over these. The pro-inflammation, so causing inflammation. This encourages very specific immune and some non-specific immune responses. That's going to be interleukin and tumor necrosis factor, okay? Interleukin 1 specifically. Interleukin 10 is anti-inflammatory, and so that's going to kind of dampen down specific and non-specific immune responses. Serotonin and histamine are important. These are vasoactive mediators, vasodilators, so increasing blood vessel size, or vasoconstrictors, decreasing the diameter of blood vessels and permeability. And so these guys are really important. Serotonin and histamine particularly, okay, which those are the only ones here. And so these are important. Then we have our growth factor cytokines that help to regulate lymphocyte growth and activation, and that's going to be your IL-7 and erythropoietin. And we'll look at erythropoietin later on. That is directly tied to development of the blood cells.